We have a question in the chat as well. Go for it, Ron.
Ah, sorry, yeah, I saw that. But just while we're on this topic in such a detailed way, I found myself very interested in a group of cells in the Jourdain et al. Paper, which I think might be very similar to these passage 8 cells. I don't know how they'd map in the way you describe, but it's obviously very interesting to go back and look at the lineages that are presented there with this in mind. I found it very stimulating. I hope that's coherent to at least to Alex. Maybe not to many people. But do you see what I'm saying? That in the Jourdain paper, there's a very beautiful description of a gliogenic lineage, which I think is going to be very similar to what you're discussing here. After all, what happens in
well, but here we don't have but we don't have glia here, right? Ron, we don't have glia here because I did the single cell RNA sequencing at DEV8.
No, but that's fine. That's true. But what you were just discussing is not this experiment. You were just discussing the passaging experiment.
Yes.
And the person, yes. Nivan or Novin Palavkan. Nice to meet you, by the way.
Nice.
Was asking questions about the passaging.
Yes.
And I think that's actually he was getting at a very interesting question, which is what happens to the neuroepithelial stem cell state if you keep it at 20 micrograms per mil of FGF over this long period? And what happens is it matures. It goes through a neurogenic phase, and that's very defined, and then it becomes this later cell that you call gliogenic. And I just wanted to say that I think you can see that in the UMAP or the T-SNE plot in Figure 1, I think, of the Jourdain et al. Paper. I'd be happy to talk.
Yeah, but we see also here so the answer is that at the early passages we have neuroepithelial stem cells, but then these cells progress, and there is also the progression in their fate commitment and their fate potential. So I said that at the beginning we have organizer cells, then in passage 4 we have neurogenic cells or neurogenic radial glia cells. Passage 8 these cells express genes of the later radial glia cells and gliogenic radial glia cells. So I said that there is a progression of these cells in vitro, which also recapitulate the in vivo, the progression of the radial glia cells in vivo too. This is what we show in this paper 2020. I didn't show here, but we have a beautiful projection that where each passage corresponds to a specific phase of the human brain development.
Nicola,
yes.
Let me interrupt you for a minute. Can you hear me? So, because I think this point that we're raising now is very interesting. In Figure 1A of this latest paper from Lorenz's lab in Nature, there's a description, or a graphic description of these events, and there's an arrow pointing to synchronous, I think what they call synchronous neurogenesis, and it's this event that we're discussing here that they're taking advantage of in their paper then, to look at the timing of neuronal maturation. So I think I'm not hesitating to emphasize to people how important I think it is to have a clear view of what this heat map really means. I hope that's a useful contribution. Because then all of your work on risk, right, just maps into these sort of fundamental features of the molecular timing of the system there. That's a nice summary, I think.
Yeah.
So here, I'll read this additional question from the chat here. So Pau Costa asks, based on the variability of gene expression in different patterning areas from different patients with the same disease, i.e. autism, should we distinguish the different subtypes of the disease and potential treatments based on that? Yeah. Yes. Right. If we can do that.
Well, this is a very difficult question. I don't know, of course. How can we distinguish the treatments? I don't know how we can distinguish the treatments, but for sure we can model based on this idea that we can have this differential bias of the cells, or we can I mean, it is our hypothesis. Okay, we need for sure to be careful with this to give the conclusion. We are still investigating if really there is a differential bias of this SD in terms of in terms of their ability to recapitulate pattern center states at early time point. So it looks like there is pattern genes are maybe deregulated across some different lines. Some lines may have an interventional bias, other lines may have a dorsal-caudal bias. We don't know if this is related to the disease, of course, right? But here, what looks like is promising that these transcription factors, which are patterning, involved in patterning, are deregulated across the different lines. But as I said here, in one pair they are upregulated, in another pair are downregulated. I mean, you can see MACE2 here, right? In these two pairs, it's downregulated in SD. In this other pair, is upregulated in SD. So that's why here we plot the frequency of perturbation independently of the direction. And this is what I can say. I don't think I can say more. I don't think I can say more about this. And there is also another result. Yes.
There is a. I encourage you to say more, Nicola, though, because before you go on to another result, just let's go on to the ASD result.
Yes.
Because Alex hopefully is on the line. Yes. And I think this is very interesting what you're saying, and it's directly what the question is asking. A very obviously interesting question for us to understand is how many types of ASD are there? And in Alex's previous seminar, he emphasized that there are the two different kinds which are on either side of a normocephalic
right.
control group, right?
Right.
As if there's two different ways of the tuning system being wrong. And I would be very interested to hear Alex's view about this question, really.
Well, I can also anticipate the fact that here we are not considering.
All the seminars are about cell proliferation, right? Either patterning genes or cell cycle genes, and surely that's what we're reading out here.
Right. So we already found that
enthusiastic.
So if Alex is a connector, we can he can also give an answer. But what I can say here is that it is story to compare also the normocephalic or to put in consideration this analysis normocephalic versus microcephalic. Microcephalic is also another level of investigation that we are considering to do. But which complicates also a little bit of things. So for now, we would like to consume, let's say, this result, which is still it's encouraging, it's promising, but we don't know really what means that this transcription factor are perturbed across multiple lines, but in a totally different way. So there is we found in our tree, but what I'm trying to say here is nothing related to really autism. My point is more related to the fact that we can model the early phase of individual brain development with this system, and this can be the fact that we can integrate this data with multiple lines with other public data sets is also a good approach that, in my opinion, we can be commonly used, right? Because of course the modeling disease is always tricky. what are the number of the ideal number of lines that we should use to model, faithfully a disease? Probably hundreds, right? But for sure, something that it takes time and resources, etc. So what I'm trying to show here is that we could have integrating this in vitro system with the in vivo monkey data sets. Then we could identify a potential phase of the development of the human development or human brain development, which may be altered, and then we tested this hypothesis on multiple cell lines, and that is what we did. And the last point is that in these 3 lines, we did find differential cell proliferation, difference in cell proliferation rate. In particular, the ASD were more in G1 phase, which was also in a line with the other paper from Giordano et al. And this means that they are maybe more prone to differentiate, right? Because a longer G1 may induce cells to differentiate faster. And that is, yeah.
Yeah, if I may say, that's really interesting data that it's an angle that we approach only in the report that we did on our side. That's, of course, we saw some different organoids having certain bias towards like anterior posterior, dorsocaudal, etc. And it's something that's we didn't associate directly to ASD at the beginning, and most of what we described, notably this normocephalic and macrocephalic, was more in the sense of a consequence, as we were seeing like bias towards like different neuronal lineage. And we didn't actually manage to relate that to what was happening early on, except the only thing we could associate it with was potentially this change in cell division, as we mentioned. And so it's really interesting here to look at this and know we can do it, notably like with Nicola's data on the monkey patterning centers, to try to understand how this bias transcribed in each ASD line at T0. And of course, then I jump back on what Nicola was saying, that indeed we would need like way more ASD line to try to start categorizing. And it's also interesting to understand that those transcription factors, as you were showing, I think from the Cell Oracle, that they can have an impact at different degrees and different cell types, right? So that complexifies even more the categorization of how to interpret.
Absolutely, yes, absolutely, yes, exactly, yeah, absolutely, yes. And actually here I'm showing you just the lineage, the radial cell progression, the glogenic lineage, but we have the same data also for the neurogenic lineage, and you can see that. Of course, some prescription factor have an effect on the on radial cell progression, but not during neurogenesis, and vice versa. And so this is very important to show that when the lesion is functioning, is important for the disease. And of course, this data also shows that the transcription factor may have can also cause more or less severe perturbation, depends on when they are working, right? I showed you this is the real that shows that if you have a mutation of this gene, the perturbation of this gene in a different phase than earlier neural stem cells maybe, this is my speculation, may give a different perturbation severity compared to the effect of the same mutation when this transcription factor is speaking, etc. But still, you don't know, because as you can see, this transcription factor also linked to late transcription factor. So maybe a mutation at this point, the perturbation at this point actually can affect also sequential events.
We have one more question in the chat.
Yeah, I
Go
was
for
just
it.
going to say, I just want to emphasize that the question directly relates to the point that you're discussing now, Nicole. And I had the privilege of looking at this image that you showed me in some conversation we had about a month ago or so. And I remember being kind of blown away by it, and then thinking quite a bit about Cell Oracle and about the importance of these inferential tools. And that's exactly what the questioner asked about. So it would be great if you could just, maybe you can just read it. And
I'm trying to connect to the chat, but here, okay. So the chat is from Soraya. Ryan is asking, how did you build the transcription factor network? Did you use the in silico perturbation to validate the network itself? But this is a very interesting question that is more related to the bioinformatics. Yes, using Serol, basically here there are two different ways to build the transcription factor networks. Here in this cartoon, in this analysis, we are using a method which is RCIS, and then the other one, the other one is with Cell Oracle, which use different parameters. But I cannot go more in detail because it is something that the bioinformaticians are more expert than me. Probably Carlo wants to say something, but I don't feel to say more than that.
Yeah, so it integrates the Trevino. Go for it, Joe, please.
Okay, so
Joel is our mascot here, so it is the guy who made
hello,
this
Nicola.
analysis. Hello, Joel. Thanks for intervening.
So in the network that you're seeing right now, this network is based on the bulk expression and in the disease association tables that we had. So in this network, we used a SysTarget, which is a tool that uses motif enrichment analysis when you provide it a list of genes. So given this list, we generate this regulatory network, and then we validate if we observe correlation of the expression in our expression dataset. Sorry. And then in the Cell Oracle part, the gene regulatory networks are created based on the ATAC dataset that the Trevino dataset contains. So in this case, the regulatory networks are created from the co-accessibility of the chromatin peaks, and then expression model is fit using the regulations observed in that ATAC-seq dataset. So we have both approaches: the motif enrichment analysis of the bulk and disease associations tables, and then we have the co-expression and co-accessibility of the multiomes in the Cell Oracle.
Oh, thank you so much. Thank you.
Okay.
May I ask if you saw kind of some kind of overlap between those two types of network computation as well when you did this analysis between the Trevino and what you did from the bulk data? Did you saw some kind of overlap?
So the first observation is that for some of the networks that we get from the motif enrichment analysis, so from this first one, we don't observe even co-expression of the core transcription factor and the target genes in the bulk expression. This could be for a series of reasons, but like maybe this transcription factor and target gene regulation is not taking place in the bulk data that we have. But then when we compare it with the Cell Oracle, you can in Cell Oracle we do some more in-depth network analysis, and we do check the centrality of the transcription factors in the different cell types across neurogenesis and gliogenesis. And in there we can observe that many of the genes that were very central, that connected many diseases in the bulk expression and in the disease associations, are also very relevant, sometimes for a specific cell type, and sometimes for the whole neurogenesis or for the whole gliogenesis.
Right. This is something that, of course, because of the time I didn't explain. But here we calculate. Here we define the role in the for each transcription factor in the regulatory network. So you can see, for example, some genes are highly connected. Some transcription factors are highly connected in all the cells. Other transcription factors are highly connected in some cells, like this EGR2, but poorly connected in other cells. And this again explain the different function of transcription factor across different cells, across different cell states of during neurogenesis.
So
I
maybe
just want
if
to
people,
compliment you on
yeah,
this discussion, guys. Yeah.
go for
I
it.
think it's great to talk this way and just to encourage people to take part. You know, these lineages are being increasingly well described by many different tools. And here in this group here, we're very focused on the behavior of cells in a dish. That's reflected in the title of the seminars we were all attending. But I'm very struck by how when people sequence adult brain, they're coming up with all these same cell types. But what's different between one person and another is the representation of a given cell type. And I think that we can go from there into a very interesting discussion, which has both a sort of informatic component to it or a biological component. But just to come back to the issues that we've talked about today, it seems to me that both seminars point to the value in trying to understand exactly the locus of the origin of the effect that you really want to study if you're thinking about human brain diversity or blood diversity. Is that a reasonable summary, Carlo? Anyway, I've really enjoyed listening to the whole discussion. Yeah.